Bismillahirrahmanirrahim. Dear students, have a nice day. Hope you will be fine best of your health. This is Dr. Sajid Mahmud Rao, Division of Science and Technology, University of Education, Lahore. Today, we are talking about a brief overview of the natural products, its applications, and pharmaceutical commercializations are the same. Let's see what are the natural products. Natural products are the organic compounds produced by the living organisms, plants, insects, marine organisms, and marine animals. Different kinds and classes of the natural products are produced within these sources. Literature survey shows that more than 300,000 plants have been reported, but only 3% of these plants are subjected for the structural elucidations of the target compounds. In Pakistan, God has gifted with enormous varieties of medicinal plants, but only 20% of these medicinal plants have been evaluated for their therapeutic effects. So, there is a need of coordinated and well-organized work in the field of natural product chemistry. Natural products are used in our daily lives, breakfast, lunch, and dinners. Similarly, most of the natural products are used in the formulations and evaluation of pharmaceutical dosage form that includes nutraceutical food supplements, Ayurvedic products, and pharmaceutical drugs. More than 50% of the pharmaceutical drugs have their origin from the natural product chemistry. This is the schematic presentation of the natural products, which gives information how the natural products are commercialized into pharmaceutical dosage form on the basis of in vitro and in vivo studies and on the basis of phytochemical screening. So you can see in the first step, the plant is collected on the basis of availability of the plant. Literature survey report which gives information that either no work has so far been carried out on this genus and species. Third, biological, enzymatic, and pharmacological importance. The next step is plant identification. In this step, the plant is submitted to the botanist, which confirm its genus and species and allow us for further process. The next step is shade drying. Here, the whole plant material is placed in shade for drying purpose, which may take some weeks or some months. The next step is chopping and grinding. In chopping and grinding, the whole plant material is chopped and grinded by using pestle mortar or by using crusher and grinder. The next step is soaking. In this step, the powder material of the plant is dipped in methanol by using methanol polar organic solvents because our target is to extract maximum number of phytoconstituents from less to high polarity. The next step is filtration. The filtration is carried out by using Whatman paper or simple paper or porcelain cloth. Here we get the final methanolic extract in the form of filtrate. The methanolic extract is now converted into solid mass by using rotary evaporator. The next step is fractionation. Here in this step, distilled water and methanol is added in the solid mass to get two layers in organic solvents. Fractionation is carried out by using organic solvents, and that organic solvents are anhexane, chloroform, ethyl acetate, and butanol. So here in fractionation, we get 4 different fractions on the basis of organic variety system, and that fractions are anhexane fraction, chloroform fraction, ethyl acetate fraction, butanol fraction, and aqueous fraction. Now all the fractions are subjected to phytochemical screening. Phytochemical screening is a qualitative analysis which gives information about the secondary metabolites and confirm how many and which kind of the secondary metabolites are present in each fraction. On the basis of phytochemical screening, now the fraction which have maximum number of phytoconstituents and secondary metabolites are subjected to in vitro and in vivo studies so that we can search more active fractions in the sense of biological active, enzymatic active, and pharmaceutical importance fraction. Here, this fraction is further selected for the column chromatography. In column chromatography, column silica is used as a stationary phase and different combinations of the organic solvents are used as a mobile phase. This technique is used for the isolation and the separations of the organic compounds or the phytoconstituents on the basis of the organic purity system. The next step is thin layer chromatography. This is a technique which is used to confirm the purification of the compounds. This technique is carried out by using the different combination of organic solvent. If we have a single compound upon our TLC card, it means the compound has been pured. Now, this compound is submitted for the spectral techniques, which includes IR, MAS, proton NMR, C13 NMR, HMBC, HMQC, NOE, COSY, and MALDI techniques. All these spectral techniques are useful for the structure elucidations and the structure terminations of the organic molecules. Now, the next step is nutraceutical, Ayurvedic, and pharmaceutical dosage form. Here in this step, the more active phytoconstituents and active crude herbs are subjected for the formulations and evaluations of nutraceutical, Ayurvedic, and pharmaceutical dosage form. In the next step, different kinds of excipients are used as a preservative and flavoring agent, which are used for the purpose of taste masking and giving the shape and giving the weight of the organic pharmaceutical dosage forms. So you can see in the final step, the pure active phytoconstituents as well as crude herbal extract, which is more active, both are subjected for the formulations and evaluation of the pharmaceutical dosage form. And these pharmaceutical dosage form may be syrup, capsules, sachet, suspension, and tablet dosage form, which are commercialized on the basis of activity ingredients. Finally, you can see here the pharmaceutical finished products from herbal extracts and phytoconstituents. These pharmaceutical dosage form are tablet dosage form, capsule dosage form, and syrup dosage form. All these three pharmaceutical dosage forms are more appropriate and have more marketing values. Natural product chemistry is at the intersections of many fields that include ecological biochemistry, taxonomy, entomology, combinatorial chemistry, chemical synthesis, microbiology, molecular biology, biotechnology, biochemistry, and pharmaceutical sciences. So these fields interlinks with the natural product chemistry. Now come to the real topic, that is alkaloids. When we see the statement of these molecules, we observe all the phytoconstituents that have nitrogen atom within the heterocyclic ring system, generally known as alkaloids. These are the secondary metaboloids. Secondary metaboloids are those substances which are used for the cure and treatment of different diseases, and these can be taken on demand. The primary metaboloids are those substances and chemicals which are very necessary for the development and the reproductions of the living cells. So, the secondary metaboloids are not necessary for the development and for the development and the productions of the living cells. When we see the structures of these molecules, you can see the caffeine, which have 4 nitrogen atoms in the molecule. The 2 nitrogen atoms are present within the hexacyclic ring and 2 nitrogen atoms within the pentacyclic ring. In the second molecule, that is nicotine, in the nicotine 1 nitrogen atom is present in the aromatic ring and the other is present in the 5-member ring. The third molecule is ephedrine. Ephedrine does not include in the genuine alkaloids molecules because the nitrogen atom is present outside the ring system. However, the physical characteristics are generally similar to those of the alkaloid molecules. The alkaloids are soluble in organic solvents, but their salts are all soluble in water and alcohols.